Hello, everyone. Welcome to today's webinar. I'm Britt Erickson, Senior Editor at CNEN, and I will be moderating today's event. This webinar is the first of a two-part series on protein quantitation, opening doors to a whole new view of biology with tandem mass tag, TMT, multiplexing workflows, being sponsored by Thermo Fisher Scientific. In Part 1 today, we will hear about mass spectrometry sample prep for new and experienced users. Be sure to also register for Part 2 of this series, LCMS Practical Guidelines for Robust, Accurate, and Meaningful Quantitative Proteomics, which is scheduled for November 9th at 11 a.m. Eastern, 8 a.m. Pacific Daylight Time. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of your screen and type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by Thermo Fisher Scientific, a leader in the life sciences industry. The company is committed to providing systems-wide proteomics solutions to laboratories worldwide to address the challenges of biological research. With industry-leading LCMS solutions, consumables, and workflows driven by the Thermo Scientific Orbitrap LCMS, the company enables scientists to be on the leading edge of research. Thermo Fisher Scientific's leadership and scientific network, publications, and pioneering solutions enable broader and deeper analyses to decipher the complexities of biology. During the presentation today, we will hear from Ryan Bomgarden, a senior staff scientist at Thermo Fisher Scientific, who is responsible for the development of products for the proteomics reagent market. Ryan holds a PhD from Stanford University, where he worked on biochemical characterization of the checkpoint kinase ATR DNA binding activity and signaling in UV-sensitive human cell lines. Ryan joined the mass spectrometry reagent group at Thermo Fisher Scientific in 2009. In this role, he has developed numerous products, including the tandem mass tag TMT 10-plex reagents for relative quantitation of proteomic samples, HeLa protein digest standards for LCMS QC, MS cleavable cross-linkers for protein structure analysis, heavy protein IVD kits for production of stable isotope-labeled proteins, and ActiveX probes for kinase, GTP-ACE, and serine hydrolase inhibitor profiling and detection. And with that introduction, I will now hand it over to Ryan to tell us more about how to prepare samples for mass spectrometry.
Thank you for that introduction, and welcome to the first part of a two-part webinar on mass spectrometry sample prep for quantitative proteomic workflows. Mass spectrometry has become a predominant technique in biological research by enabling the identification, characterization, and quantification of proteins on a global scale with high accuracy and precision. Shotgun or bottom-up proteomics is a common strategy to identify proteins from peptides created from proteolytic digest. But the complexity and large dynamic range of proteins in samples greatly influences the depth of coverage one can achieve, while the quality and the consistency of sample preparation affects the reliability and the accuracy of the results. In this presentation, I will provide an overview of sample prep challenges specifically related to MS proteomics and describe multiple strategies to reduce sample complexity and improve the reproducibility of protein sample preparation and peptide quantitation before and during mass spectrometry analysis. Most protein biology researchers are familiar with the typical Western blot workflow, which includes a gel-based separation, a transfer step to a PBDF or nitrocellulose membrane, and detection of proteins using primary and secondary antibodies, followed by an imaging for data analysis. Mass spectrometry is also similar in that we separate proteomic samples with an HPLC and analytical column. We detect those digested peptides using a mass spectrometer, but then we typically analyze the raw data post-detection with database tools such as database searching software and quantitation software. Although Western blotting has not changed much in the past 35 years, protein researchers are looking for new and complementary methods that are less dependent on antibodies, provide a comprehensive analysis of proteins and PTMs, have improved quantitative accuracy, and can complement the global genomic data that they have with other technologies. Fortunately, the barriers to adoption of LC-MS as an analytical tool to study proteins has significantly decreased over the past decade through improved instrumentation and software. However, unlike the Western blot workflow, mass spec sample preparation involves numerous additional steps beyond cell lysis, which induce more variability and decrease reproducibility in MS measurement. These include cell lysis, reduction, alkylation, digestion, and cleanup. In addition to cell lysis to extract the proteins, you typically have to sonicate cells at a denaturing buffer to shear the genomic DNA to remove that later in the sample preparation. Mass spec sample preparation also requires reduction alkylation of cysteine involved in disulfide bridges before enzymatic digestion. And finally, it requires cleanup to remove salts in the sample and buffers and other contaminants which may interfere with LC-MS analysis. Some of the sources of variability that can occur during sample prep include incomplete sample lysis, reduction, alkylation, and digestion, sample loss among the different steps, as well as the sample cleanup step at the end, poor reagent quality that can also lead to off-target effects during, for instance, alkylation of the cysteines, and finally, just the fact that there's more steps during mass spec sample prep alone is one source of variability. And overall, this basically decreases the reproducibility from sample to sample, leading to high coefficients of variation when you do quantitative measurement. Another big issue with mass spec-based sample preparation is that there's no standard workflow for preparing samples, even for the same sample type or even among the same lab. A common workflow is to perform in-gel digestion, where proteins are first separated by SDS page or 2D electrophoresis. Advantages of this method include a reduction in sample complexity because you are spreading the proteins out by molecular weight. Because of that size effect of the gels, you get a knowledge of the general molecular weight of your protein. Due to protein staining of the gel, you can get an estimation of the protein amount. And finally, the gel itself, gel electrophoresis, removes a lot of other sample type contaminants such as detergents and salt that may be present in lysis buffers. However, there are also several disadvantages to in-gel digestion for quantitative proteomics, and that includes that it's not really scalable for large sample amounts. There's only so much sample you can apply to each well. It's very time-consuming, so it's an extra step to run the gel, stain it, destain it, as well as prepare the sample for mass spec analysis. The actual gels themselves are more costly, and also the peptide extraction from in-gel digestion is quite variable and is different depending on where you cut the bands out of the gel from different sizes or different regions of the gel. In contrast, in-solution digestion methods are more scalable and more reproducible, but are not compatible with a wide variety of cell lysis components, including salts and detergents. In addition, there's a wide variety of in-solution digestion sample preparation techniques, including magnetic bead separation, filter-assisted separation sample prep, such as FAST, solvent precipitation methods that include acetone precipitation, methanol, or methanol chloroform mixtures, and finally, urea-based lysis. Each one of these in-solution sample preparations has different pros and cons, which we'll go through in the next slide. So shown here, with the exception of using chaotropes such as urea or guanidine hydrochloride for protein preparation, most in-solution digests use a combination of SDS detergents, and heat for protein extraction, solubilization, and denaturation. Urea-based sample prep methods are still quite popular due to their simplicity and low cost. However, urea alone does not extract proteins from cells and tissues as efficiently as SDS-based buffers, and can also modify protein lysines through a process called carbamylation, which inhibits tryptic digestion and increases sample complexity. Of the device-assisted methods, filter-assisted sample preparation, or FAST, is the most widely used. Although this method is reported to have high protein extraction efficiency and reproducibility, the method involves numerous washing steps which require long centrifugation times to remove SDS, urea, and other small molecules before digestion. In addition, FAST is difficult to scale for different sample amounts and volumes based on the device size. Detergent removal spin columns and solvent precipitations, which is used in the Pierce mass spec sample prep kit, are two alternative methods to remove SDS with high efficiency in a single process step. But of these, the precipitation method is the most reproducible and scalable. As shown in the LC chromatograms on the left, the sample preparation method used in this kit can be easily scaled from as little as 10 micrograms up to 5 milligrams with extremely high digestion efficiency as determined by the number of peptide miscleavages, which are typically between 3 and 7%, depending on the instrument used for detection. This method also provides complete protein reduction and alkylation with high reproducibility. In addition, the kits come with a standard protein at a known concentration that can be used as a digestion indicator to assess digestion efficiency across multiple samples. As mentioned previously, even with the most sophisticated modern mass spectrometer, it is still not possible to detect and identify and measure all the peptides from a complex protein digest sample. Unlike absorbance or fluorescence, which can be used to directly correlate signal of your sample to relative abundance, mass spec is not inherently quantitative due to a wide variety of issues, including a wide dynamic range of the abundance in the sample, the sensitivity limits of the instrumentation you're using, differences in the ionization potential of different analytes, and this can also lead to ionization suppression if two analytes happen to co-elute off the column at the same time. And lastly, there's a lot of missing information from run to run, or even as you're sampling the sample, including using data-dependent acquisition methods, which only maybe measure the top 20 most abundant analytes in a certain duty cycle. And also, you may be matching with the spectral library. And again, if you haven't seen the peptide in previous runs, you may be missing it in the future. But there are some solutions. Reducing sample complexity makes samples more palatable for the mass spectrometer, allowing more measurements to be acquired, which ultimately increases the quantitative precision. However, the most accurate quantitation by mass spec requires the addition of stable isotope labeled internal standards, which can be used to generate standard curves of known concentration ranges or compare multiple samples relatively within the same LC-MS run. So how do we reduce complexity? Well, there are a wide variety of techniques that can be used to fractionate samples at the protein and peptide level. As mentioned earlier, gel-based electrophoresis methods are a powerful method to separate proteins based on their size and charge. Other techniques can be used to enrich various proteins through their subcellular localization, enzymatic activity, or immunoaffinity. Today I'm going to highlight a few key methods, including abundant protein depletion, PTM-based enrichment, specifically phosphopeptide enrichment, and high pH reverse phase fractionation chromatography, which can dramatically increase the depth of protein coverage with low impact on sample variability. The first technology I'd like to talk about is abundant protein depletion. Unlike simple unicellular organisms such as yeast or mammalian cell lines, which have a dynamic range of protein abundance up to 6 orders of magnitude, the dynamic range in plasma is greater than 12 orders of magnitude. To put this in perspective, this is a vast difference, is equivalent to trying to identify one star among all the stars in our entire galaxy. Although this may be possible if you know where and what to look for, in global plasma proteomics, it is quite challenging to identify any proteins besides albumin and antibodies, which make up approximately 80% of the total plasma protein composition without depletion. These proteins represent a huge barrier to detect the lower abundant proteins, which are commonly associated with human health and disease. There are a lot of products and formats that can be used for abundant protein depletion, ranging from straight albumin affinity using Cybercron blue dye resin to antibody-based immunodepletion of the top 2, top 12, or top 14 most abundant proteins. However, the immunoaffinity-based enrichment methods are the most specific and reproducible. As you can see on the gel on the left, without depletion of serum albumin, antibody heavy and light chain fragments dominate the gel. But after depletion, these proteins are noticeably reduced so that other less abundant proteins can be detected. These immunodepletion resins are quite efficient at removing greater than 99% of their target protein as determined by LC-MS targeted quantitation, and this level correlates well with results from ELISA. That being said, even after depletion, albumin and IgGs are still some of the most abundant proteins in the sample due to their large dynamic range. The top 2 and top 12 aborting protein depletion resins are available in convenient single-use spin column format. One advantage of this format includes quick processing time of up to 10 microliters of plasma using only a microcentrifuge. In contrast to the more expensive LC column formats, there are no potential for sample carryover from run to run, and no decrease in performance which is observed after multiple LC amino affinity column regeneration. As shown on the right, without depletion, only 962 peptides can be identified by LC-MS in the sample, most of which map to the same abundant proteins of albumin and IgG. However, after depletion of the top 12 proteins, we identify nearly double the amount of unique peptides, mapping to many lower abundant proteins. Although individual samples are processed using different devices, these devices are highly reproducible. As seen here, there is greater than 90% overlap in protein identifications after plasma depletion using different devices. The depletion reproducibility across the 3 top 12 columns resulted in an average coefficient of variation of approximately 12%, compared to a CV of about 8% from technical variation alone. In addition, using label-free quantitation to measure protein relative abundance after depletion had high concordance among replicants, as shown by the scatter plot to the right. The next technique that can be used to reduce sample complexity is phospholipid enrichment. Similar to the detection of low abundant proteins in plasma, detection of phosphopeptides is difficult as unmodified peptides dominate the LC-MS spectrum, which leads to poor detection. Although there are some chromatography techniques which can partially enrich phosphorylated proteins, the most common methods enrich modified peptides through affinity to the negatively charged phosphate groups. These include immobilized metal affinity chromatography, IMAC, of which an example is iron NTA, metal dioxide chromatography, which the most common example is titanium dioxide, and phosphospecific antibody-based affinity chromatography, of which is used commonly for phosphotyrosine enrichment. Unfortunately, phosphor groups are not the only negatively charged functional groups found on peptides, so there can be some significant nonspecific binding of unmodified peptides to these affinity resins. Specifically, carboxylic acid groups found on amino acids and peptide C-termini can compete for binding, reducing the specificity and enrichment yields using these techniques. This is especially true for titanium dioxide resins, which typically require a blocking buffer containing lactic or other organic acids to reduce nonspecific binding. Recently, we re-optimized the buffer formulation and protocols for our iron NTA IMAC and titanium dioxide enrichment kits. As shown in the graph, we have significantly improved the binding specificity for both resins to greater than 95%. In addition, our new IMAC kit can enrich up to 150 micrograms of phospholipid from only 5 milligrams of mammalian cell culture digest, nearly tripling the yield compared to the older method. Our reformulated dilution buffers are also mass spec compatible and can be removed by speedback, which eliminates the need to clean up the samples using offline C18 cleanup or trap columns, which can result in loss of hydrophilic phosphopeptides. A common debate in the literature is which method is better in enriching phosphopeptides. However, both iron NTA and titanium dioxide actually enrich different subset of phosphopeptides. As shown in the Venn diagram, there's less than 60% overlap in unique identified phosphopeptides enriched using each method. Previous reports have noticed a slight bias for enriching multiple phosphorylated peptide species using iron NTA. However, we observe a slight bias favoring titanium dioxide with our newer buffer formulation. That being said, our iron NTA kit spin columns have approximately 1.5 times the loading capacity of our titanium dioxide kits, which makes them a little bit better suited for larger scale enrichment. Ultimately, the choice depends on the application and the peptides of interest. Next, I want to switch to peptide fractionation. As mentioned earlier, sample complexity is a key challenge for detection of low abundant protein and to quantify them. Offline separation of protein digest using strong cation exchange or SCX and high pH reverse phase chromatography have become popular methods to increase protein identification. For SCX fractionation, samples need to be exchanged into a loading buffer for binding, and elution is typically performed with increasing concentrations of ammonium formate or salts, which require additional cleanup before LC-MS analysis. In contrast, for high pH reverse phase fractionation, acidified protein digest can be directly loaded onto the columns and eluted with increasing concentrations of acetonitrile under basic conditions. Another advantage of high pH reverse phase fractionation is that it is more orthogonal separation mode to the low pH chromatography used for analytical LC-MS C18 columns. Similar to the top 2 and top 12 abundant protein depletion resins, we have developed a convenient single-use spin column format for high pH reverse phase fractionation. The steps involve first equilibrating the columns and conditioning them using buffers provided in the kit before sample loading of up to 100 micrograms of protein digest under low pH conditions. Then the columns are washed to remove any salts, and then we switch to a high pH buffer before elution. Bound peptides are then eluted in a stepwise fashion using increasing concentrations of acetonitrile ranging from 5 to 50%. One thing that can be observed in the chromatogram shown here is how orthogonal the separation mode is compared to low pH fractionation. In the top chromatogram, you can see a high pH fraction of a 7.5% acetonitrile concentration that's been subsequently analyzed in a low pH mode by LCMS. In the second chromatogram in the middle, we see a low pH fractionation that was done on these columns and then subsequently also separated in a low pH mode using LCMS. And on the bottom is an unfractionated lysate for comparison. As you can see, the peptides that are pre-fractionated in the low pH mode are not distributed across the LC gradient, in contrast to the high pH mode. This distribution of peptides in the high pH fractionation mode results in better sampling during LCMS, leading to more peptide identification. Although these columns use a step gradient for elution of peptides, there is still excellent fractional resolution. Approximately 70% of each peptide is found within a single fraction, with less than 25% found in more than one. When comparing unique peptide identifications, you can also see there is comparatively low overlap observed in adjacent fractions. This results in more unique peptide identifications as the instrument is sampling fewer redundant peptides from a less complex mixture. Fractionation using these devices is also highly reproducible. Here are 3 chromatograms of the 12.5% acetonitrile fraction from 3 different separations. As you can see, the base peak chromatograms are nearly identical. When looking at the individual peptides from different proteins, we also observe less than 10% variation in relative intensity among replicates. Since the initial complex sample is spread among different fractions with unique subsets of peptides, more peptides can also be loaded onto the analytical LCMS columns, which enables detection of more low-abundant peptide species. Overall, in comparison to unfractionated samples shown in the black bar, there's a roughly 2-fold increase in unique peptide and protein identifications from the combined high pH reverse phase fractionation samples shown in red. However, this depth of coverage does require 8 separate LCMS runs, which can decrease sample throughput when comparing multiple biological conditions. Next, I'm going to talk about TMT reagents, which are an isobaric tagging technology that can help improve both sample throughput and quantitation reproducibility. So the TMT workflow is depicted on this slide. First, we can start up to 6 to 10 different conditions. These would be your biological samples. And after performing your mass spec sample preparation, which involves your cell lysis, reduction, alkylation, digestion, you then would label with the different TMT tags. These TMT tags are amine reactive tags that label on the peptides on the amino terminus as well as lysines, and each tag has the same nominal mass, but when combined, they do not increase the sample complexity. The samples are then combined in equal molar mixtures, fractionated, for instance, using high pH reverse phase, and analyzed by LCMS. During the MS of the peptides, you basically select all the same peptides from all the samples, which is then triggered for MS sequencing. During tandem MS, you get the peptide sequence ions for sequencing, and in the low mass region of the spectra, you also generate unique reporter ions based on the different isotope distributions of the reporters that can be used for relative quantitation. Some of the advantages of sample multiplexing is that you get your time back. You can basically combine multiple biological samples into one and do one single fractionation and LCMS analysis so that you can basically analyze up to 10 samples at once. Another advantage is that there's fewer missing values. Since all the samples are combined into one LCMS run, there's virtually all the peptides that you see can be quantified, and therefore there's fewer missing values from run to run. Because there are 10 channels, one of the channels can be used as an internal control or a pooled sample. This reference sample verifies performance and can basically be used for comparison across multiple experiments, especially if you have more than 10 samples and you want to use it as a bridge channel. And lastly, with multiple channels, you can do multiple comparisons to have improved statistics. And for this talk, I want to focus on that we can actually incorporate biological replicates within the same set of isobaric tag samples to compare multiple conditions in the same time. In addition, you can use the multiple channels to do more sophisticated experiments, such as dose response curves, time courses, or look at multiple tissues or sample types, or even subcellular fractionation samples. However, there is an issue when quantifying isobarically tagged peptides in unfractionated complex protein digest mixtures. Since isobaric tag reporter ions are measured after peptide fragmentation in MSMS, if there is coelution of abundant precursors with target peptides, it can result in interferences in the reporter ion region, distorting the ratios during quantitation. Even when using a narrow isolation width of, for instance, 1.2 AMU, co-isolation of an interferent peptide still presents a challenge to accurate quantitation using isobaric tags such as TMT, ITRAC, and others. If the contaminating peptide is also isobarically tagged, the resulting ATD-MS2 spectra contains reporter ion intensity from both the target precursor and the interference. This distorts quantitation from its true value and results in a marked decrease in both the accuracy and precision, depending on the degree of co-isolation. The real change in protein expression, blue, therefore becomes masked under a screen of reporter ion intensity from the contaminating peptides shown in red. Ultimately, this can lead in a bias in the relative quantitation ratios towards 1, resulting in lower reported fold changes if the interferences are abundant. But interference can be reduced and even eliminated with the proper sample preparation, LC separation, and instrument methods. So in the top graph, we have an experiment where we're going to demonstrate the problem with interference using complex samples. As you can see, we have in the blue graph a TMT-labeled BSA sample that's mixed at 16 to 8 to 4 to 2 to 1 to 2 to 4 to 8 at fixed ratios, and we are going to spike in a constant background of a complex sample of a HeLa digest that was unlabeled with 8 of the 10 TMT channels equally mixed at 1-to-1 ratios. And as you can see in the bottom graph, we would expect the ratio of the BSA, when we quantify it correctly, to basically follow that twofold dilution series as expected. However, with channels that contain interference, such as shown here 128C, when the interference is much higher than the target peptides you want to quantify, it distorts the ratio, resulting in less precision and less accuracy. However, shown in the light blue bars at the very end of blow up, you can see that there's a new method that we've enabled on the Orbitrap Fusion and Lumos platforms that allows us to reduce or eliminate the interference. And this is called SPS-MS3. Since label-free analysis compares samples from separate LC-MS run, quantitation can also be dramatically influenced by column load, the spray instability, and changes in chromatography performance. In fact, due to the high reproducibility of isobaric tagging quantitation, the CVs are typically twofold less than label-free techniques, as shown here in the graph, with CVs typically around less than 5%. So basically, I've gone through a lot of different types of samples, and this shown here again is the TMT workflow. We've talked about using sample preparation techniques for reproducible sample preparation up front of quantitative proteomics. We've talked about using isobaric tags such as the TMT reagents to label multiple samples to combine them for doing concurrent quantitation of multiple samples within a single LC-MS run. We've also talked about high pH reverse phase fractionation and phospholipid enrichment to reduce sample complexity. Next, I want to talk about some new tools that are really important for normalizing protein samples before LC-MS injection, as well as TMT multiplex labeling. So I'd like to introduce two new peptide quantitation assays. The first is the thermoscientific Pierce fluorometric peptide quantitation assay, or FPQ for short. And the second is a thermoscientific Pierce colorimetric peptide quantitation assay, or CPQ for short. The fluorometric assay actually uses a fluorescent dye that fluoresces upon labeling the peptide amino terminus. It's a very quick assay. The reactions are complete in less than 5 minutes and has excellent linearity, as I'll show in a minute, and very good sensitivity. Unfortunately, since the peptide amino termini are labeled with this dye, it's not well suited for TMT-labeled sample quantitation. In contrast, the CPQ assay uses indirect copper reduction chelation chemistry similar to the microBTA assay. Because of this, it does require a slightly longer incubation time. It is an indirect assay that we typically can complete in 30 minutes or less. It also has a very linear response and is also more sensitive than microBTA. and is actually not really recommended for single peptide analysis. So shown here on this slide are the standard curves for two different proteins. One's a protein BSA digest, and the second is bacitracin with the FPQ assay. As you can see here, the standard curves show excellent linearity and sensitivity down to less than 5 micrograms per mil, and they can also be used to measure simple and complex mixtures. The FPQ assay is also significantly more sensitive than absorbance at 214. The CPQ assay is also a more excellent assay for measuring peptide concentration and is more linear than microBCA over a greater dynamic range, especially at the low microgram region. The CPQ assay has actually got 3 to 4-fold increase in signal to noise and a fold increase in sensitivity compared to this very classic assay that's been used for protein quantitation. And this is the assay that we recommend for complex peptide mixture quantitation. As I mentioned previously, the CPQ assay can measure both labeled and unlabeled peptides, and shown here on the graph is the response from a TMT-labeled HeLa digest in blue compared to an unlabeled HeLa digest in red, and that the response is similar with or without a TMT label. However, in the fluorescent protein quantitation assay, since the amino termini are blocked by TMT upon labeling, the fluorescent dye cannot react, and therefore it is not suited for measuring the protein or measuring the peptide concentration. Both assays are actually quite consistent and comparable. In this graph, you can see the quantitation of peptide digests from 12 different cell lines, and the peptide concentrations were determined using both the CPQ and the FPQ assays. Each sample was analyzed in triplicate, and the concentration of each digest was calculated from a standard curve using a protein digest assay standard that is provided in each kit. As you can see, both assays give a very similar response for all these different sample types. One of the biggest benefits of measuring the peptide concentrations after digestion or cleanup is that you can normalize the amount of peptide loaded onto analytical columns for LC-MS. This results in much higher run-to-run reproducibility and can even lead to more peptide identification if combined with optimized LC conditions and instrument methods. Shown here on the graph on the top are the number of peptides from using only protein-based quantitation to normalize your samples, and then you go through your sample preparation. And due to the variances that I talked about earlier between digestion efficiency, sample cleanup, and the amount that you inject on column, there's a much broader variability in the number of peptides that can be identified. However, using these peptide assays to normalize the amount of peptides that you have post-sample prep cleanup and before LC-MS analysis, you can get very reproducible protein group and peptide identification. So I've described a number of sample prep technologies and methods and kits that can be used to decrease sample complexity and increase quantitative reproducibility while maintaining sample throughput. So I'd like to end with a workflow slide where I put all of these together. So this is a generic TMT workflow that was performed where we have replicate samples that were labeled with different TMT channels. Shown here we have 3 samples labeled that were untreated, 3 that were treated with insulin, and 3 that were treated with IGF-1. And the last channel was used as our pooled reference channel of all of them. So the samples were serum starved and treated with the different conditions. The proteins were isolated and prepared using the sample prep methods I described previously, involving the denaturing, the reduction, alkylation, and digestion with trypsin. Each sample is then independently labeled using the TMT tags and then quantified using the peptide assays before equally mixing them. From these samples, we also did, the combined samples, we did high pH reverse phase fractionation to look at the relative abundance of all the proteins in the sample. In addition, we did separate phosphopeptide enrichment followed by high pH reverse phase fractionation to do in-depth coverage of the phosphoproteome for relative quantitation and signaling. As shown here with the TMT Templex reagents that are combined with this phosphopeptide enrichment workflow and high pH reverse phase fractionation, we had a very large increase in the number of peptides that we can identify by LC-MS. So in a total of 16 runs, we're able to quantify over nearly 14,000 phosphopeptides and from more than 6,000 proteins with an FDR of less than 1% and a minimum of 2 unique peptides per protein. This workflow also shows the compatibility of phosphopeptide enrichment procedure with the TMT10 labeling technique. And one thing I wanted to highlight was the relative quantitation of the reporter ions from the TMT labeled peptide shown here on the bottom graph. And as you can see, using peptide normalization and TMT quantitation, you can achieve extremely reproducible results among your replicate. So in summary, I have covered a wide variety of sample preparation kits and standards to improve sample quality and reproducibility. Using peptide fractionation upon protein depletion and phosphopeptide enrichment, you can identify and quantify more relevant proteins in your sample and identify more biomarkers that may be relevant to the human disease condition. And finally, using peptide quantitation techniques such as the new colorimetric peptide assay, fluorescent peptide assay, in combination with TMT labeling, allows you to quantify to get the best accuracy and precision in your quantitative proteomic workflow. And with that, I first would like to thank my colleagues at Thermo Fisher Scientific in the Protein Biology MS Reagent Group and the Thermo Scientific Proteomics Marketing Group. And I would also like to thank my collaborators for graciously sharing their samples, data, and expertise. And lastly, I'd like to thank you for your attention, and we'd be happy to answer any questions that you may have.
Thanks, Ryan. It is now time for the Q&A part of this webinar. As a reminder, to ask a question, please type your query into the Q&A box. Okay, it looks like we have several lining up in the queue. All right, Ryan, can you please explain more about the normalization method?
Yeah, sure. I think peptide normalization after your sample prep is probably one of the key problems that I see with the majority of sample prep today. I mean, although you may measure the protein concentration of each sample prior to reduction, alkylation, and digestion, there's a lot of variability in the cleanup, especially using homemade devices. And the numbers that you get when you're doing either protein identification, and especially when you're doing quantification in the instrument, is highly dependent on the amount of sample that you load on your analytical column. So I think that the ability to use a peptide assay post-cleanup to normalize the amount that you're putting into the instrument dramatically improves quantitation, even if you're doing label-free quantitation, and especially if you're doing more sophisticated workflows such as using the TMT quantitation.
Okay. How well does this method work with blood with a large protein background, like albumin?
Right. So albumin is a really challenge, and so I typically recommend using depletion. Although you can do protein quantitation in blood without doing depletion, having fewer sample preparation, fewer techniques used to prep your samples will obviously lead to better reproducibility. However, you only be able to identify a few hundred to a couple of 500 to 600 proteins, even using the most sophisticated mass spectrometer. So by dealing with the depletion of the most abundant proteins, you can get much better depth of coverage and hopefully able to quantify the markers that you're interested in at the lower level. I will say that the products that I highlighted in the talk today are for human plasma and serum only. They do not work for mouse plasma or serum because the antibodies that are mobilized on the resin for depletion are specific to human proteins.
Okay. I think you may have answered some of this in the beginning, but do you recommend measuring peptide concentration before or after the C18 cleanup? And are there any reagents that interfere with the peptide assays?
Yeah, that's a very good question. I typically recommend measuring after cleanup, and there's a reason for that is that there are some reagents that do interfere, small molecules and chemicals that do interfere with the different peptide assays. So let's start with the fluorometric assay. So that assay has an amine reactive fluorescent dye, and so any amine-containing buffers, such as that contain a primary amine, such as ammonium bicarbonate or tris, which are common buffers used for digestion, would quench the tag. So therefore, would react with the tag and give background, I would say, not necessarily quench the tag. So because of those buffers being commonly used for digestion, I recommend measuring the peptide concentration after cleanup. And also, like I mentioned earlier, there is a lot of variability in the cleanup, so you want to kind of capture that by measuring right before you put it in the mass spec. That being said, the colorimetric assay does work well, and you can measure in tris or other digestion buffers. However, that chemistry in in those colorimetric kits is based on the same chemistry of the micro BCA. It has a slightly different chelator that gives much better signal to noise and sensitivity at the low end, which is great for peptide samples because you can use a lot less material than you would for a micro BCA assay. However, there are things that do interfere with the BCA assay. So anything that has your reductance, so if you don't do a precipitation and if your reducing agent is still present in your sample, that could interfere with the peptide assay even if it's in the digestion and you've diluted it out. The other thing is that some of the ammonium bicarbonate and triethylammonium bicarbonate, so TEA, and some bases also interfere and give a slightly elevated signal using the colorimetric assay. So although the colorimetric assay can be used, I think, for protein digestion, you have to be a little bit careful on the amount of certain reagents. It is highly tolerable to a lot of interferences, but majority of the other things like acetonitrile or other additives you may add to improve digestion, both assays are compatible. And the last thing I wanted to reiterate is that the fluorescent assay cannot quantify TMT-labeled peptides because the amino termini of the peptides are blocked by TMT. So only the colorimetric assay can be used for measuring TMT-labeled peptides.
Okay, and which instrument is best for TMT quantitation, and can lower resolution instruments be used?
Yeah, so as I mentioned in the talk, the SPS MS to the third method is the best method that can be used to improve the accuracy as well as the reproducibility of your TMT quantitation. So that requires an instrument that's capable of doing MS to the third, so a trap-based instrument. And the high resolution required that's actually provided by the Orbitrap instruments is necessary to separate the 6-plex tags into the 10-plex. So you need to have a resolution of at least 30,000 M over Z 200. So therefore, the Orbitrap Fusion and Lumos are the two instruments that have that acquisition method enabled. The Q Exactive series of instruments have Orbitraps as well, and the Elite and Velos Orbitraps can also have the resolution requirements to separate a 10-plex. But for low resolution instruments, you can actually still do TMT quantitation, but you would be restricted to using the 6-plex. Those tags have unit mass Dalton resolution, so therefore they can be easily measured on a low resolution instrument, such as a Velos or a regular ion trap or even a QTOF instrument.
Okay. I'm going to go back to the colorimetric peptide assays. The question is, can we use the thermo nano drop for measuring colorimetric peptide assay?
Yeah, that's a great question. It turns out that we currently recommend to use a plate reader, so UV Vis spectrophotometer plate reader. The nanodrop is capable of reading the wavelength. The wavelength is it's a different colored chelator, so it's a slightly different wavelength than BCA. I believe it's 488 instead of being 562 nanometers. So the nanodrop is capable of reading it. However, the formulation of the final reaction, the buffers do contain a little bit of acetonitrile. And so what happens on a nanodrop when you're only using 1 to 3 microliters of your reacted sample in the buffers that contain the chelator, what happens is that the evaporation of the reaction happens really quickly on the nanodrop because of such a low volume. So because of that, it's very difficult to measure all of your samples reproducibly using a nanodrop. We are working on a slightly different formulation that has less organic that may be more compatible with the nanodrop, but it does work. But just to note that you do have to move pretty quick to measure once you apply the sample to the pedestal to measure the sample because the evaporation will actually affect your reading over time. So I definitely recommend plate readers over the nanodrop, but the nanodrop does have the ability to read that wavelength.
Okay. This question is about immunodepletion kits. It says, do the immunodepletion kits work for mouse plasma as well?
Yeah, I kind of covered that a little bit earlier in a different question, and unfortunately they do not. The antibodies that are mobilized to the resin only recognize human proteins, so they do not work for mouse.
Okay.
Yeah.
Okay, great. What are the most common issues you see with MS sample prep today?
The biggest one I see is that people aren't monitoring sample loss through their workflow. I know a lot of people, in order to get reproducible results, you do have to basically check things as you go. So you want to, obviously monitor the amount of protein that you're extracting from your cell lysates or tissues. You want to monitor the peptide that you get at the end. Some of the other issues that I see that are quite common is that people are kind of overdoing things. So they're adding more reagents to try to make things go faster, or they think more is better. And unfortunately, adding too much alkylation reagents results in over-alkylation, and adding too little or too much trypsin can either result, too little would result in high miscleavages, and too much is just a waste of reagents. So basically, what I basically see is that there are a lot of homebrew protocols that I think within the lab people are highly familiar with and have optimized. But when new users are trying to basically do mass spec sample preparation, they sometimes don't necessarily understand all of the reagents that are in the workflow, and some of the concentrations that they use are excessive, and that leads to basically poor results downstream.
Okay. And I just refreshed, and we've got lots more questions, so hang in there. If I use both an iron NTA and a titanium dioxide for phosphopeptide enrichment for the same sample, is there a % increase in the peptide ID that I can expect?
That's a good question. So if you use them separately and actually do separate LCMS runs, we typically see the overlap between the identified peptides to be roughly around 50%. Now, that is just an identified peptides. If you actually do an algorithm like ASCORE or PhosphoRS to localize the site, the percent overlap may decrease a little bit. But typically you see about 30 to 50% orthogonality with those two products. There are some protocols that actually use them sequentially, and we found that when you use them sequentially, there's really not a lot of material left after doing the iron or the titanium first to do the second enrichment. So what ends up happening is that the capacity of the resin is kind of scaled for, let's say, a 500 microgram to 1 milligram sample load. And when you basically do them sequentially, what we could recommend doing is taking the flow through from, for instance, the iron NTA column. You could then dry it and then resuspend it and apply it to the titanium dioxide column sequentially. However, in our hands, we haven't really seen a dramatic increase doing that method. We actually just see a much better increase in identifications using them separately to basically pull out different types of phosphopeptides.
Okay. Here's another question on depletion kits. Are there other depletion kits other than the top 12, like a top 20 or more?
Not currently, but we have some of those in development, and so stay tuned. But right now, we only have top 2 and top 12. There is a top 14 product, I believe, available from Sigma, but we do not have anything beyond the 12. Okay.
Does the abundant protein kit work for plant samples? Can it remove, say, Rubisco?
Unfortunately not. The antibodies that are immobilized to the resin are specific to the top 2, which are albumin and IgG, or top 14 human proteins. So they are species-specific just for humans.
Okay.
But a Rubisco depletion column is a great product idea, and I don't think there's many companies that are selling that. So we'll have to keep that on the list.
Okay. This one has to do with software. I'm running an experiment for 200 plus samples and will be using TMT quantitation. Do you have a suggested software for the statistical analysis of these samples, as a Proteome Discoverer 2.1 doesn't support this form of analysis?
Yeah, that's a good question. So a lot of people, so I will say that 2.1, you're able to process and bring together quite a number of files. I don't know the upper limit, but I know I've brought in dozens of files together from multiple runs and able to bring them together to look at the signal to noise across all the different samples. You can also do study design and change your variables. So for instance, if you use one TMT channel for one particular sample in one experiment, if it's in a different channel in the second experiment, you can actually pull the quantitation together in a study design. So I think it does facilitate the viewing of the data, but for statistical analysis, you'll have to use some other type of stats program for fold changes. But it does give you the raw data and exported. And I will say that I won't be able to go into much detail on the software, but the second part of this webinar series, will be much more in depth on MS acquisition methods for TMT labeled samples, as well as phospho TMT labeled samples, phosphorus-rich TMT labeled samples. And in that, we'll actually have a speaker go through the Proteome Discoverer workflow and cover data analysis specifically for TMT.
Okay. Can you further comment on the method to minimize compression phenomena using the fusion?
Yeah, so that method is the SPS MS3. Again, it'll be covered in much more detail in the second part of the webinar. I just wanted to highlight, introduce it in this particular method. So the way that method works, and I apologize, I was missing a slide in my presentation that I usually cover. The way this works is during the TMT tags fragment using HCD, so using higher collision-induced dissociation. So what we do is this was developed by Graham McAllister in Steve Geege's group, and he now actually is an employee of Thermo working on improving these algorithms. But the way it works is that instead of doing a higher collision energy MS2, we actually do a low collision energy CID during MS2. So what that does is actually clean up the spectra by basically doing a mild fragmentation of the peptides. And so if there is a co-isolating interfering ion that has the same, very similar mass, upon doing low energy CID, it actually will separate the interfering mass from the target mass that you're interested in. And then during MS to the third, there's an algorithm. This is the SPS MS to the third. It was originally called multi-notch in the publications from the Geege group. And what it does is it selects between 5 and 10 of the most abundant precursors in the MS2 scan to combine them and to inject them in the orbit trap. So it goes through HCD and then it all gets injected in the orbit trap. And basically all 10 precursor ions from the MS2 scan are combined and then fragmented for one MS to the third scan. So what this allows you to do is clean up your spectra by going through another MS event. And you also get your reporter ions are basically quantified from your MS3 data, and your peptides are identified from your MS2 data. So you basically get kind of the best of both worlds between ID and quantitation of the reporter ions. With this method, you can also control now. They've updated the new tune files, which will be covered in the next talk. We'll also cover some advancements in these algorithms. Basically, you can adjust the windows and the widths or the numbers of notches that you select so that the quantitative accuracy is even much more improved than the very first version. But that's basically how the method works. The one caveat is doing an MS to the third approach, you do lose some signal, right? So you do need to load a little bit more material on column, and we also recommend using high pH fractionation and longer LC gradients so that you can basically maximize and the identification rates to try to offset some of the issues that you're going to have with basically duty cycle, because you are doing a third scan. The instrument is slower than doing, for instance, just an MS2 method.
Mm-hmm. Okay. Is there any trick during immunodepletion to only remove the top 12 and not the other interesting proteins that may be associated with the high abundant proteins?
That's actually a good question, and unfortunately, no. So that is one issue with doing depletion, is you could also co-immunoprecipitate or co-deplete a protein of interest. Albumin especially is known to be kind of a reservoir in the plasma with a lot of protein-protein interactions. So that is an issue with using depletion to go up to lower abundant species, is that you could also remove some of the other proteins. I will say that the depletions are quite reproducible. So if you do remove something, it's probably being removed very similar in all your samples. But that is one caveat of doing depletion. Mm-hmm. One trick I do recommend is that for after depletion and post-depletion, but prior to tryptic digestion, is to actually boil the samples. And the reason I mention this is there actually are a lot of proteases in the blood, so endogenous proteases. You can imagine the ones that are involved in clot formation, but they're like thrombins and whatnot, but there are also other additional proteases. And so what happens is post-depletion, you can reduce alkylate your sample, you add trypsin, but you basically get a lot of semi-tryptic peptides or non-tryptic peptides in your digest. And what that does is actually increase the complexity of the plasma even further. So you're not just digesting with trypsin, but there's other enzymes in the blood sample that are actually active during that digestion and cause a lot of other peptide products. So I do recommend post-depletion to boil the sample before reduction alkylation, and that basically will denature all the endogenous proteases and make your sample complexity much less. Do not boil or denature before depletion, because obviously if the proteins are unfolded, they will not deplete, because the antibodies have to recognize the native form of the protein.
Okay. When using the TMT 10-plex, which tag should be used to label the internal standard? We used TMT 126, but I saw you use TMT 131.
That's a good question. It doesn't really matter, I think, in theory. One thing I will say is that if you've used TMT before, it does come with a data sheet, and the data sheet includes some isotope impurities that are present in the individual tags. So although the tags are highly purified commercial-grade reagents, greater than 93% pure typically, the isotope purity is also greater than 99%. There is, in the 126 tag, there is obviously a lot of carbon-12 and or undoped carbons, and there is a naturally occurring carbon-13 in all of our molecules. So therefore, there is a 1%, 1.3% amount of carbon-13 isotope in naturally occurring carbon precursors. So therefore, there is about 8 to 9% impurity of carbon-13 in the 126 tag that spills into the 127 channel. And so that's easily corrected using software. So in Proteome Discover and other softwares, you can do what's called an isotope correction, and you enter in the impurities, which we determine here in the factory and provide them to C of A, and then you can subtract out those impurities. The one thing about using your pool channel with the 131 at C, the impurities actually spill into 132, so you don't have to subtract them out. But really, it doesn't really matter which channel you use for your internal standard, as long as you apply your correction factors. And if you're using a high-resolution instrument, for some channels, they don't require correction, because as long as you get up to 4 or 5 decimal places, they don't necessarily spill exactly. For instance, the N isotopes, the N tags don't spill into the C tags and vice versa. So if you're using the 6-plex, you don't even really need to correct that much. So anyways, I guess my answer is it doesn't really matter. It's kind of a personal preference. But there may be an application where you may want to use one over the other. And many researchers actually maybe run one set of 10-plex using samples 126 through 131, and then they'll invert their sample order just to kind of take care of some sample prep or run bias. But since the tags are identical, there's a lot of papers that are published with both iTrack and TMT to show that no matter which tag you use, the tags all behave identically.
Okay, here's another one. How much sample is required for quantitating labeled TMT peptides?
That's a good question. So the format, there's actually a few formats for TMT. So there's actually a pre-aliquoted format. It contains 0.8 milligrams of tag per tube, and that can label 100 micrograms per sample. So if you have a 10-plex, for instance, now you've combined it, and now you have 1 milligram of TMT labeled sample. And after cleanup, you'll probably have at least 80% of that. So that's a lot of peptide material. So for most LCM analyses, even using a 50-centimeter column, you pretty much only need to load 1 or 2 micrograms on. And if you have your replicates within your TMT, you may only have to shoot the TMT sample once or twice to get your technical replicates on the instrument. So you're basically labeling a lot of sample. And so unless you're doing phosphopeptide enrichment, in that case, 1 milligram is perfect. I mean, some people typically like to use 1 to 2 milligrams for phosphopeptide enrichment, because after enrichment, you only end up with about 10 to 20 micrograms of peptide sample. So that's the right amount to basically have some material for the peptide assays, as well as for doing your LCMS analysis. So for that format, I definitely recommend it for doing phospho TMT workflows. We are launching a new product format that contains 0.2 milligrams, so 4 times less tag, and that product actually should be on the shelf in about 3 to 4 weeks. So it's in manufacturing right now. It was introduced to ASMS, and it'll be highlighted at the HUPO meeting coming up in Taipei. But basically, this format is actually going to be a 96-well format, so it'll have either 80 vials of TMT 10-plex or 96 vials of the TMT 6-plex. And there'll be individual vials that are screw-capped and stable in a stable packaging format. and that'll allow you to label only 25 micrograms of sample. So we've kind of scaled down the labeling reactions, and therefore you basically aren't wasting as much TMT, because I know a lot of people try to make their own aliquots or reconstitute the tag and then dry it down and freeze it. And unfortunately, the TMT reagents, although they're extremely reactive towards amines, they're also very reactive towards water. So temperature is not that big of an issue, but water and moisture is the #1 killer of TMT. And so what happens is that if customers don't handle the product correctly or store the product correctly, the tag will hydrolyze, and therefore they will get poor labeling of the amino termini and the lysines, and that'll lead to variability in the quantitation. With the recommended sample amount and protocol, we typically see greater than 99% labeling of the amino terminus and lysine residues. But if the tag has gone bad or been mishandled, then you will see underlabeling. That being said, in our formats that we provide commercially, in the pre-aliquoted formats, we have a 1-year guarantee on the stability of the reagent.
Mm-hmm. Okay, I see we've made it to the top of the hour, and that's all the time we have. And thank you again, Ryan, for your fascinating presentation. And thank you, participants, for being a great audience. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. And if you haven't done so already, be sure to register for Part 2 of this series, LCMS, Practical Guidelines for Robust, Accurate, and Meaningful Quantitative Proteomics. It's scheduled for November 9th at 11 a.m. Eastern, 8 a.m. Pacific Daylight Time. Thank you On24 for technology and production services, and thank you Thermo Fisher Scientific for the sponsorship that made this interactive webcast possible. For CNN Webinars, I'm Britt Erickson. Goodbye.